Hello, welcome to Dynamical Modeling Methods for Systems Biology. My name is Eric Sobey. I'm an associate professor at Icahn School of Medicine at Mount Sinai in the Pharmacology Department, and I'm going to be the instructor for your course on Dynamical Modeling Methods for Systems Biology. Thank you for signing up, and I look forward to working with you as we teach about different methods that are used in systems biology to analyze and implement dynamical mathematical models. In this first video, the topics that we're going to cover are illustrated on this outline. We're going to discuss the overall course goals. We're going to discuss the specific biological topics and mathematical topics that are going to be covered in this course. And then we're going to review the overall structure of the course and how we're going to perform grading and assessment in this course. The overall goals of this course, Dynamical Modeling Methods for Systems Biology, are threefold. First, we want to teach contemporary methods that are used in systems biology for dynamical modeling. Second, we want to teach methods for mathematical analysis of biological systems and simulation output. In other words, you implement a model, then you get some output from the model. So what sorts of approaches do you use to analyze that output and gain more insight into the biological system through that analysis? And then third, we want to demonstrate how dynamical mathematical models can provide novel insight, the type that you cannot get if you're only doing experiments. So the combination of experiments with dynamical mathematical models is going to provide novel insight that is very difficult to get, or sometimes impossible to get, just from the experiments themselves. So what do we mean when we talk about dynamical mathematical models? Well, I think it's helpful to divide different computational approaches, different types of mathematical models, into two categories. And I like to differentiate between statistical, or what I call top-down models, versus dynamical, or what I also call bottom-up models. So what do I mean by that? Well, the approach you take in a top-down model can be summarized as follows. You begin with a data set, and often a very large scale data set, the kind that you might get in a genomics experiment or a proteomics experiment. Then you use statistical methods to find patterns in that data set. And then once you've used statistical methods to find patterns in that data set, you can generate predictions, and the predictions are based on the structure that you've uncovered within the data. And so some of the keywords that you can associate with this top-down approach are things like network analysis, gene set enrichment analysis, clustering algorithms, principal component analysis, or partial least squares regression. Now I should note that these top-down approaches to doing mathematical modeling, these statistical models, are not the focus of this course here. These top-down approaches were taught in a Coursera course that was offered by my colleague from Mount Sinai, Dr. Avi Mayan, and Dr. Mayan is planning to offer this course through Coursera again in the next few months. So if your primary interest is in these types of models, learning clustering or learning principal components, I would encourage you to take a look at Dr. Mayan's course. Our focus here is going to be on a different category of model, what I call dynamical models, or what I also call bottom-up models. So the approach here is sort of the opposite of what we saw with the top-down model. With a bottom-up model, you begin with a hypothesis of biological mechanism. Once you have this hypothesis, you write down some equations that describe how the components in your biological system interact with one another. Then you run simulations to generate predictions for what would happen under different conditions. And some of the keywords that are associated with bottom-up models are things like ordinary differential equations, tools of dynamical systems to interpret the output, methods for parameter estimation, partial differential equations, and stochastic models. Now in this course we're not going to be able to cover everything, so we're going to focus on the first two keywords in here. Models consisting of systems of ordinary differential equations and tools of dynamical systems in order to interpret the results of these simulations. And this dichotomy of statistical models versus dynamical models has been discussed in the literature and in several review articles, including one of ours. This is an article that my colleagues and I published that describes a course that we developed at Mount Sinai School of Medicine. And this Coursera course has evolved in many ways from this course that we teach at Mount Sinai. So if you're interested in learning more about this dichotomy of statistical versus dynamical models, I would encourage you to check out this article. And there's other articles in the same category, in the same class that also discuss distinctions between different categories of models. I think it's worthwhile to review the general structure that you frequently see with a dynamical modeling study. That should give you some sense of what the steps are that are involved and what kind of insight you can get from this type of study. So usually in a dynamical mathematical modeling study, you begin with some idea of the mechanism, of what happens biologically. So I've just illustrated this with a simple example here where you have some biological species B can get converted into some other species C, and this protein, or the species A, might regulate conversion of B to C. For instance, A could be an enzyme that catalyzes conversion of B into C. Once you know something about the mechanism, or you hypothesize something about the mechanism, you write down some equations describing how the different components in the system interact with one another. And this is an example where we have two equations, and these are in the category of ordinary differential equations that describe how a particular system, how the components of a particular system evolve with time. Once you've written down the equations, then you write a program to simulate those equations. And these are the first few lines of a program that does such a simulation. The programming environment we're going to use to write these programs that simulate our ordinary differential equation-based models is called MATLAB. And so we're going to have several lectures that introduce you to the MATLAB programming environment before we show you examples of dynamical mathematical models that are implemented using MATLAB. Once you have your program, you run simulations with your program that simulates the equations. So this is solving the temporal evolution of one species and another species. And you can see that the black one is oscillating with respect to time, and the red one is oscillating with respect to time, although the actual shape of the oscillations is different between the one that's represented in black and the one that's represented in red. So then after you've run these simulations, how do you make sense of them? Well, then you often use tools of the field of dynamical systems to analyze your output. And one of the things that you might do frequently is you might vary some parameter, and then you might look to see how your output changes as you vary this parameter. And this is a case where you, under low values of the parameter, you can get high values of output alternating with low values of output that would be analogous to these oscillations that we're seeing over here. But then with higher values of the parameter, you only get a middle value of the output. And so in this case, the oscillations have ceased. So by running these types of simulations where you vary a parameter and you analyze the output, you can gain insight into how the behavior of the system changes under different conditions. So this is the general structure of a dynamical modeling study, from mechanism to equations, to a program that simulates the equations, to simulation results, and then to analysis of the output. Now let's discuss some of the logistics of this course. The format that we're going to take is as follows. It's going to be a 7-week total course consisting of approximately 25 lectures, each lecture being approximately 20 minutes. If we take 25 lectures, divide that into 7 weeks, we see that we're going to post somewhere between 3 and 4 lectures per week on average. At the end of each lecture, we're going to provide you with one or more self-assessment questions. This is a way for you to think about what you've learned in the lecture, try to answer the question to assess for yourself how well you've understood the material in the lecture. And then the overall assessment is going to be based on 5 homework assignments that are going to be given after each lecture block. So the 25 lectures are going to be divided into 5 blocks. Each of these 5 blocks is going to be associated with a homework assignment. And then passing the course is going to depend on completing these 5 homework assignments. The skills that we wish to teach you in this course are the following. First, we want to teach you to use this programming environment called MATLAB to be able to perform data analysis and data visualization. Next, we want to teach you how to develop models consisting of systems of ordinary differential equations. And throughout this course, we're going to abbreviate ordinary differential equations with ODEs, like that. Then we want to teach you how to implement ODE models in MATLAB and how to run simulations with these models. And then finally, we want to teach you how to analyze ODE models using the tools of dynamical systems analysis. And one of the arguments we're going to make as we go through this course is that models can be useful for understanding several different biological processes. And some of the biological processes that we're going to discuss in this course are the following. We're going to talk about glucose oscillations in yeast. We're going to talk about kinase signaling pathways in mammalian cells. We're going to talk about regulation of the cell cycle. This is a case where mathematical models have been extremely successful and extremely important in understanding whether or not cells want to divide or whether they want to stop dividing. And we're also going to talk about mathematical models of electrical signaling in neurons. That's another classic case where mathematical models have been really critical for getting a quantitative understanding of how the biology works. And the overall goal is to provide you with the tools necessary to apply these types of models to your own questions of interest. So some of you might be interested in some of these specific biological questions, but many of you are, might have some interest in some other biological mechanism or some other biological pathway. But the hope is that if we teach you the tools, you can use the same tools to whatever system it is that you're the most interested, that you have the most interest in. How are we going to perform assessment and self-assessment in this class? Self-assessment is going to be performed by providing you with 1 to 2 questions at the end of each lecture. And I'm going to introduce the question, I'm going to give you some time to think about it, and then after you think about it and answer it for yourself, I'm going to go through the explanation. So this will be a way for you to think about what you've heard in each lecture and try to apply the concepts that you've heard, and then you can see for yourself how well you understood the material that was covered. Overall assessment in this course is going to be performed through the homework assignments. The homework assignments are going to require you to perform simulations with dynamical models. Because this is a course on teaching you methods, on teaching you the sorts of approaches that are used in systems biology, the way that you're going to really learn these approaches is by doing the simulations yourself. So the homework assignments are going to ask you to perform simulations with dynamical models. And the general format we're going to use is not to make you write the entire model from scratch. We're going to provide you with some MATLAB code that does one thing that might run a simulation of a particular biological system, and then we're going to ask you to adapt it, to modify it in order to do something else. And then we're going to provide you with assessment questions that are designed to verify, first of all, that you've implemented the model correctly, and also that you can get some biological interpretation from the results that you've obtained with that mathematical model. So we want to test both your programming skills to implement it correctly, and that you can get biological interpretation from your results. And this is to sort of reiterate what I just said on the last slide. Homework assignments are designed to reinforce concepts that are discussed in the lectures. And the assignments should require you to demonstrate 3 things. 1 is technical competence. Can you actually implement the model correctly? 2nd, quantitative skills. Can you get a reasonable quantitative interpretation of your simulation results? And 3rd, can you use the model, can you use the simulation results to obtain new biological insight into the particular problem and into the particular system that you're looking at? So thank you for listening. That concludes our first lecture, and I look forward to working with you in this course, Dynamical Modeling Methods for Systems Biology.